Hello, good afternoon. Presenting today is Mustafa Said Kilong, a student from Industrial Engineering, medical laboratory student. Yes. Today we will talk on some questions or subtopics in immunology. There is a question asking T helper cells how they support the B cell activation. We have isotype switching and its regulation. We have T follicular helper cells and their importance. We have also effectiveness of a vaccine. What makes a vaccine effective? Yes, we can proceed further and we talk about the first as how the T helper cells support B cell activation. Yes, we've known the antigens that activate the B cells with the help of the T cells are known as the T cell dependent antigen, and they include the foreign proteins. Yes, they said the proteins cannot uh, cross-link multiple B cell receptors due to their lack of repetitive and identical epitopes. Yes, and this activation of the B cell, it has some three signal steps. The first steps we know that it is the antigen recognition and binding by the B cell. Then the second one, um, here we have the B and the T cell interaction. Then finally, we have the cytokines released by the T helper cells that stimulates the B cell. Yes. Further, we can also talk that you can see here that uh, they are named such as because they are unable to induce immune response in organisms that lack T cells. Yes, the B cell responses to the antigens takes multiple days. It takes long time for it to respond to antigen or to foreign antigen. Yes. Let us talk about the T cell independent activation. Uh, Here, once the B cell receptors bind to the foreign antigen, the antigen is taken up into the B cell through the receptor mediated endocytosis and presented to the T cell as peptide pieces in a complex with MHC2 molecules on the cell membrane. The T helper cells, typically the follicular helper cells, recognize and bind these MHC peptide complex through the T cell receptors. Yes. Um, we've known that following the binding of the T cell receptors to the MHC2 peptide binding, we see that the T cells expresses the surface protein, which is the CD40 ligand. Yes, as well as cytokines such as the interleukin 4 and interleukin 2. Also, interleukin 5 they are produced. We've seen that the CD40 ligand serves as a necessary co-stimulatory factor for B cell activation by binding the B cell surface receptor uh, to CD440, yes, which promotes the B cell proliferation. Yeah, and again, we can see that immunoglobulin class switching and somatic hypermutation, as well as sustained T cell growth and differentiation. This is all the effects of the CD4 CD4 ligand, what it does to the B cell. Yeah, the T cell derives cytokines bound by the B cell cytokine receptors as well also to promote the B cell proliferation, immunoglobulin, the same as the class switching, the somatic hypermutation, as well as to guide differentiation. After the B cell receives these signals, they are considered as activated. Yeah, we have seen that, and we can see here in the image, we have seen the T helper cell. First of all, they say that the first step, they say that there's antigen recognition by the B cell, antigen recognition and binding by the B cell receptors. Now you have seen there is a foreign antigen here being recognized and it binds to the receptors of the B cell. After binding, the antigen travels into the cell, the B cells, whereby it will trigger the MHC peptide complex. Mm. Here, this complex um, will help in this form. The B cell will process the CD40 in the co-stimulatory receptor. Here is the receptor where we see the CD40. Yes. After we can see that. they have said something about uh, we have seen something very important talking about the dendritic cells. Before these T helper cells are activated, we have seen that the dendritic cells process antigen in form of the MHC peptide, whereby the T cell get activated. You see, the dendritic cells form a complex of the MHC2 peptide complex and the T helper cells receptors recognize and binds to it. After, we can see here that the T helper cells which is activated comes and binds with the antigen here, and we have seen with the help of the CD4 and with the plus the CD40 ligand. Yeah, these T helper cells will release cytokines, whereby triggering the B cells to form the cytokine receptors. We can see we have the cytokines at the interleukin 2, 4, and 5. Now the T cells the T dependent B cell activation. So once activated, the B cell participate in two steps differentiation process that yields both short and live plasma blast for immediate protection and long life plasma cells and memory B cells for persistent protection. The first step is known as the extrafollicular response. This occurs outside the lymphoid follicles but still in the SLO. During this step, activated B cells proliferate and may undergo immunoglobulin class switching and differentiate into the plasma blast that produce the early and weak antibodies, mostly of the IgM. Yes. We've seen the second step consists of the activated B cells entering the lymphoid follicles and forming a germinocenter, which is a specialized microenvironment where the B cells undergo extensive proliferation, immunoglobulin class switching, which is the isotype switching, and affinity maturation directed by the somatic hypermutation. This process is facilitated by the T follicular helper cells and the follicular dendritic cells within the germinocenters and generate both high affinity and memory B cells for long life plasma cells. Yes, we can see the ISO switching and its regulation. We've known that the ISO switching can also be called the immunoglobulin class switching, or it can be called class switching or a recombination. Uh, Yes, we know that the isotype switching is a process that allows a B cells to change the type of antigen it produces without altering its antigen specificity. So antigen specificity, yeah, remains the same. It means that the variable region of the antibody which determines the antigen binding remains unchanged during the class switching. Only the constant region of the antibody heavy chain is altered, allowing the antibody to interact with different immune effectors, e.g., for example, the complement protein and the factor C receptors without changing its affinity for the antigen. So we can see here we have seen that the heavy chains, the variable chains are these ones, and the epsilons, the epitopes are this one. Means that let us go a little bit behind. They say that the antigen specificity remain the same, only that the variable region of the antigen of the antibody remains unchanged during the class switching, and only the constant region of the antibody of the heavy chain is altered. That means that this is the variable region which remains unchanged, and here the changes occurs in this region here. Yeah. So before I go further, first of all, let me talk about this isotype classification. We have known the variable region, which is the VDJ, which is also it is the DNA, and we have the mute which is expresses the IgM, and we have the delta which expresses the IgD. We have class delta 3, class delta 2, and delta 2b. We have also the epsilon and alpha. Yeah, and we can see here that the removal occurs only in this. Why do we say that? Why is it occurring only on the gamma on the on the delta? I mean, on the delta 3. They say that when there's a bacterial infection that triggers the delta 3, this means that the DNA is removed from making the B cell uh, able to make the immunoglobulin. So mostly the changes occurs from the milk to the delta 3, whereby we can see that the gene in the heavy chain locus of the IgG expresses the B cell. We can see that the transcription has occurred, and the part that is being removed or spliced is gone. Yeah, you can see the steps and mechanism that we have initially antibody production. The naive B cells initially produce IgM and IgD antibodies after the VDJ. As we have seen before, recombination during the early development. Then after that, we have activation, whereby upon encountering an antigen, the B cell is activated and begins to proliferate. It then receives signals through the CD40 and the cytokine receptors, and often modulated by the T helper cells, particularly the T follicular helper cells. In this form here, there is class switching. The activated B cells undergo class switching to produce other antibodies isotype like the IgG, the IgA, or the IgE. Yes, the constant region of the antibody heavy chain changes, but the variable stays the same. Yes, the antigen is specificity. We can go further. We see that there is gene rearrangement after the class switching, or during the class switching, the segment of the antibody heavy chain locus are deleted and the gene segment encoding the new isotype constant region are rejoined. So in this case, this facilitated by the class switching recombination, a process involving a double-stranded breaks at the conserved switch region near the constant region gene segment that is of the delta. We have activation induced deaminase, which is converts the cytosine basis in the S region into the uracil, which is excised by the DNA glycolysis, and the resulting abasic site cleaved by the endonuclease. After this, we can see that the intervening DNA between the S region is deleted, allowing the constant region of the different isotype to become connected to the variable. The DNA ends are rejoined through the non-homologous end joining, completing the switch. So you can see the fifth, we know that the antigen diversity after class switching, the B cell produces antibodies of different isotypes. We have the IG1, IG2, IgA1, and IgE, which with the same antigen specificity but different effector function. The heavy chain locus in humans, the order of the heavy chain exon is as this one. We know that the IgM, IgD, you have IG3, IgG3, IgG1, we have IgGA1, we have IgA1, we have IgG2, we have IgG4, we have IgE, and IgA2. Yes, we can talk about the mechanism of the class switching. We know that class switching occurs through the class switch recombination, where the portion of the heavy chain gene locus are deleted and the constant region of the antibody is changed. Activation induced cytidine deaminase plays a central role by inducing a double-stranded DNA breaks at the switch region near the constant region genes. This leads to the deletion of the intervening DNA, allowing the gene segment for different isotypes as IgG, IgA, and IgE to join with a variable region. The DNA ends are rejoined through the process called the non-homologous end joining. Yeah, we have these are the rules of the isotype switching. We can see that they're switching to specific isotypes. The order of the constant region immunoglobulin locus determines the isotypes that can be generated, like the IgM, IgD, IgG, IgA, and IgE. For example, after switching, B cells may produce IgA1, IgG2, IgA1, or IgE, each with distinct effector but the same antigen specificity. We have also regulation. In this process is tightly regulated by cytokines and signals from the CD40 interaction with the CD40 ligand with T helper cells. These signals activate the transcription factors like the AID, which are essential for initiating the CSR. We have also the epigenetics and chromatin remodeling. We have DNA methylation and histone modification helps regulate the accessibility of the switch region, ensuring that the class switching occurs at the correct time and to appropriate isotype. Yes, let us talk about the T follicular helper cells. You know, the thing is just going on a flow like a step. The T follicular helper cells, these are special type of CD4 T cells that help control the B cells in areas of the lymph node, spleen, and the Peyer's patches, which are places where the immune cells meet. You can see that here the naive cells, which is the immature B cells, enter when they enter the germinal cells, they become the T follicular helper cells. You know, these T follicular helper cells can also change to follicular B cells. You can see also the germinal center B cells. You see, with the introduction of the CD4 ligand, interleukin 2 and interleukin 4, you can see that the germinal center B cells can go to memory B cells and also to the PC. Yes, let's talk about the importance of the T follicular cells. We know that the T follicular cells they help in our immune. Let's talk about the induction of the germinal center responses. The germinal center are specialized areas in the secondary lymphoid organs where the B cells mature and improve their ability to fight infections. So they are important in the production of the uh, these antigens that fight infection. The T follicular helper cells are essential for the formation and maintenance of the germinal centers. They help B cells undergo affinity maturation and class switching. This which enables the more effective immune responses. Without these cells, the B cells cannot generate high affinity antibody or form memory B cells, which are key to providing long-term protection and rapid immune upon re-exposure to an infection. Yes, you see, these cells they support long-term immunity and vaccination. The memory B cells produced with T helper cells helps ensure that the immune system can quickly recognize and fight off previously encountered pathogen. And also they are crucial for effectiveness of the vaccine. They promote the generation of the memory B cells and long-lived plasma cells, both of which are needed for recall responses during the secondary infection. For example, in the studies of cholera vaccination, a memory T follicular helper cell response was linked to better antibody production and protection. So we can see that the responses to protein antigens, you can see that the T follicular helper cells are particularly important for response to protein antigens, which require T cells helper for B cell activation. For T cell dependent antigen, the T follicular cells are responsible for guiding the B cell through complex processes of germinal center formation, affinity maturation, and class switching. This in contrast, the T cells independent antigens which can stimulate B cell directly but without the robust maturation or memory responses. You can see also it helps the regulation of the autoimmunity. The T follicular helper cells have regulatory role in preventing autoimmune diseases in the immune center. They help to select against select self-reactive B cells that might generate harmful antibodies. If the T helper cells become dysregulated or overreactive, they can contribute to autoimmune conditions such as the systemic lupus erythematosus, and the Jocrine syndrome by supporting the survival of the B cells that produce antibodies against against the body's own tissues. We have aging and immunity decline. As people age, the function of the T follicular cells decline, which contributes to the immunosenescence. They create the gradual weakening of the immune system. This decline reduces the ability of the body to mount effective immune response to infections and vaccines. As older individuals may have diminished capacities to generate high affinity um, antibody or long-term immune memory. Now let us talk what makes a vaccine effective. Let's talk about it. This vaccine efficacy or vaccine effectiveness, this is a percentage of reduction of the disease cases in a vaccinated group of people compared to unvaccinated group of people. So we have some things that make a vaccine to be more effective in the body. For example, let's talk about when activation of the immune system. Vaccine introduce antigens, right, to activate B cells and T cells in the body. We know the responses of the T cells to the assisting auto immune responses. We have immune memory. The vaccine help create memory B cells and memory T cells, allowing immune system to respond quickly and strongly if the pathogens are encountered. Long-lasting protection, fake news of durable, you can see that. So we will see also safe and targeted antigen vaccine use safe version of pathogens such as the inactivated or subunit forms and may include adjutant to enhance the immune response. Herd immunity. When a large percentage of population is vaccinated, it reduces the disease spread and protecting those who can be vaccinated. Um, so at this moment, I will present you the references that you will go through and see more and see more about the subtopics that we have talked in brief. Here the references. So I thank you so much for your time. Please, um, thank you so much.